We've all played our part during the COVID-19 pandemic to keep our loved ones safe, whether it's working from home, avoiding traveling, wearing face masks, or by other means of preventing viral transmission. There have been millions of cases of COVID-19 worldwide, and infection rates continue to soar in certain parts of the world. Scientists estimate that only testing symptomatic and hospitalized cases may miss up to 80% of SARS-CoV-2 transmissions. Through the early testing of wastewater, scientists are able to detect SARS-CoV-2, identifying local COVID-19 outbreaks up to 10 days more quickly than conventional swab tests. So how does Agilent's technology work to detect SARS-CoV-2 in wastewater? The samples are transported to the laboratory for testing. 24-hour composite influence samples are collected from wastewater treatment plants. Different technologies are being used to directly detect SARS-CoV-2 viral proteins in wastewater, such as liquid chromatography, coupled with mass spectrometry, as well as the more traditional approach of PCR amplification and detection, such as qPCR solutions measuring RNA or proteins in samples to show the presence of SARS-CoV-2.
We traditionally use Agilent equipment such as LC-MS-MS, GC-MS-MS, and ICP-MS to conduct routine environmental testing. But during the COVID-19 pandemic, we decided to support our local community and use Agilent tools to test wastewater for SARS-CoV-2. We're using microbubbles from Acadium to extract the viral particles from wastewater, and the ARIA-MX real-time PCR system with the fast master mix reagents to quantify the samples. This means that it really only takes about 15 minutes of hands-on time to process these samples. We've been collecting samples from university buildings all over Southern California to monitor for COVID-19 hotspots. From these samples, we can see what percentage of the local population is shedding viral copies and help detect future outbreaks of COVID-19. Longer term, we hope that wastewater monitoring could be used to identify more conventional flu outbreaks. We're proud to be keeping university communities safer and are looking into new ways of identifying different viruses by adapting technologies and methodologies.
We have been using Agilent equipment to analyze wastewater from the Durham region here in Ontario for traces of SARS-CoV-2. Our research suggests that a viral spike in wastewater appears up to 10 days before corresponding spikes in positive clinical COVID-19 cases appear in the region. My colleague Linda can tell you more about the methodology.
We'll be using Agilent's liquid chromatography quadruple time-of-flight mass spectrometry to measure the abundance of SARS-CoV-2 proteins in wastewater. First, 24 composite samples are collected with an autosampler and then shipped to us from the local municipal wastewater treatment plants. We measure proteins bound to the particulates in wastewater, so we use ultracentrifugation to collect those particles. We then extract and purify the proteins, digesting the proteins into smaller peptides that we can sequence using data-dependent full-scan tandem mass spectrometry acquisition. Finally, we use bioinformatics software to identify proteins in the wastewater from the spectral data and to determine their functions.
This methodology is having a real impact in our region, and we are working with the Ontario government to keep our community safe. We've analyzed over 750 samples from 9 wastewater treatment plants, helping to detect early outbreaks. As part of this, we've seen over 245 peptides from 8 different SARS-CoV-2 proteins, including the membrane and spike proteins, as well as replicase polyprotein 1A, which is a very large protein made after infection and is also the protein that we detect the most often in wastewater. There are other interesting applications for the methodology we're using, and Linda's going to tell you more about that too.
Exactly. We've been thinking about future applications for this method and technology. So we want to explore using the same method for measuring numbers of vaccinated persons. We can also detect cancer biomarkers in wastewater, and we're curious to know if there is any relationship between these biomarkers and different geographic areas. Finally, we want to develop methods to look for pathogenic microorganisms that could move into a new geographic regions due to effects of Earth's changing climate.
Science and innovation are key to ending the pandemic and essential to identifying and monitoring future pandemics. Liquid chromatography and qPCR technologies will remain vital for monitoring infectious diseases like COVID-19 in wastewater samples. Agilent's diverse technology portfolio may also be applied to other infectious disease areas of research, such as pharyngitis, caused by Streptococcus pyogenes, loose smut of barley, caused by Ustilago nuda, and several others. Agilent is proud to supply equipment and qPCR reagents and instruments, which supports research scientists like Augustin, Denina, and Linda in the fight against COVID-19. Together, we are paving the way for a safer future for us all.